label. That's called isotopes. What are they? This DNA, I'm going to label use P35. Phosphate 35. And the protein, I'm going to label use S32. Because sulfide only in protein, not in DNA, and phosphates only in DNA, not in protein. So what do you do? If you label those, this is going to be the S32. This will be, I'm using another color. The DNA will be P35. Okay, once you tap it, what's going to happen? They will have a bacteria. The bacteria will be carry on the DNA sequencing coming from a bacteriophage. Okay, so this could be happened is P35. And how about the bacteriophage? The bacteriophage will also have P35 and S32 for the capsule of the source, for the capsule. So what happened is, this is bacteria, the bacteriophage, the DNA integrated into here. Then a virus is a host. Sorry, the virus is a parasite, you can say, obligated parasite. So using the chemical or the material from the bacteria to biosynthesize its own. And then the DNA has been integrated from the bacteriophage go to a bacteria. So what happened? Later on, after this invasion on the bacteria, they find P35, but no S32. However, in a new bacteriophage, they find both S32 and P35. What this means? Which means the DNA has been transferred from a bacteriophage, go to a new bacteria, go to a bacteria, which means it's integrated, or as you say, it's invaded. So that means this DNA is the genetic information has been transferred, and this message later on we call it transduction. Okay, so here is the thing. The DNA transfer, we know there are three methods right now so far. Number one, transformation. We just talk about on the right side. Number two, transduction. This is related to a bacteriophage. Number three is called conjugation. Conjugation. This is related to something we mentioned briefly in the exam 1. It's called Philly or fibril, and typically we call it a sex Philly. That's called conjugation, okay? These two did these famous two experiments to verify that DNA is a genetic information. Not done yet, okay? Not really done yet. What happened? There is something else. Continually do it. They continue to do the research. This is 1953. If you're taking genetic class or biology 101, you already know something. They say it's a most important experiment in the 20th century. Is 1953. Crick and Watson find double helix of DNA. Is that right? You know about that. This is a Nobel Prize like 1958. They are both originally from University of Indiana. They do the research in Cambridge University in England. They spent a long time based on what they did to find the structure of DNA. What's the morphology it is, what's the components of DNA. And finally, they find this double helix. The first day when they find these results, they go to a bar in England. They call it pub. Then they go there, they see all the people there, they say, okay, I'm going to buy a drink for everybody. Then everybody get a beer or wine. Then the host asks them, why are you going to do that? And they say, we have some most magic thing happens in the world. Of course, they tell them they find the double helix of DNA. Of course, nobody knows in the bar, is that right? Of course, the people in the bar, they doesn't care about it. So it seems like today, you're not going to grab a beer, go to the bar, talk about aerobic respiration, is that right? But later on, they're very excited, they come back, and they write only one page of a research notes, and that notes published in Nature. It's a very top journal, you know that, Cell, Nature Science. And they talk about this double helix of the DNA. And today we know this is double helix. Okay. Is that right? Today we know about that. Connected with each other. So the double helix of DNA. Okay, once they find this double helix of DNA, you need to verify it. It is important because it's a very magic stuff there. What is the most implication of the double helix DNA? The most implication. Because there is something story behind it. Today we know the most implication coming from double helix of DNA is semi-conservative replication. However, there is some people need to verify this. Okay, so there is another study. This is around 1954-ish, I will say. Michelson and Stahl. This guy and this guy, they are both graduate students in Caltech. Did you watch the drama called Big Bang Theory? Those are smart guys, the genius, they all coming from there, is that right? This is 1950s. Michelson and Stahl from Caltech. They did a very important research to verify the implication from double helix of DNA is semi-conservative replication. Stahl is not there anymore. He retired. He still survived. He retired. I think he's a professor at the University of Oregon and retired in Eugene. Right now he's in Eugene, Oregon. You know the Eugene, Oregon. Which one is most famous? The athletes competition for choosing the best one go to Olympic Games for the United States. Is that right? There in the Eugene, Oregon. Michelson is still there. Michelson is still a professor in Harvard University. They did their graduate study in Caltech. In 1954, they did this important research to verify how they did it. Okay. You have this. Let's say you have this one. Okay, this is DNA list. How do you verify it is semi-conservative? Okay, when you go here, replication. That's it. What is semi-conservative, first of all? They are using one of their mother bands to replication a daughter bands, and the daughter bands should be exactly the same as a mother bands. The only difference is based on the gene complementary policy. Okay, so they get these two. Now, how are you going to verify it? You have to do some of the label. Is that right? What they label? They label the bacteria N15. Not really isotope. Not really isomer. It's just a different weight. And then they grow the bacteria. They swamp them. We call it a swamp. Okay? They pour it on a medium. Then the median having all those other bacteria, which is they labeled in, is N14. Okay, then what's the difference between N15 and N14? The molecular weight, is that right? How they verify that? This is a very magic stuff. They have a large centrifuge tube. This centrifuge tube, they put something there, which is called Sicilian salt. When they pour the Sicilian salt, what's going to happen? They will, because they are different weight, molecular amount. So when you put the Sicilian salt, then you do the centrifuge. They're going to have different layer. So what they are going to separate? This guy one here is N15. And the one on the top is N14. This corresponding to this guy, and this one corresponding back to it. And the two bonds are exactly the same. So two dot mother bands, which means the two products are exactly the same. This means a replication of DNA is semi-conservative. Until that time, this is concluded. The genetic information is DNA and the structure is double helix. Okay? So then we're going to talk about the DNA. What it looks like, what it is about. So we're going to remove this one. We're going to talk about the DNA. So you know these things in your genetic class, but I just want to make sure we repeat these because it's a very important concept of the science. From the genetic information, we verify it is DNA. Transformation, transduction. We know the structure, we verify the structure. We know the end position, semi-conservative. Now we're going to talk about DNA. Okay, this is DNA structure. These slides are right here. You understand, okay? We're going to combine, talk about it. So this is DNA. Okay, first question, what is the full name of DNA? Deoxynucleic acids. It's so simple. So the first thing, 2' deoxynucleic acids. Okay, what that means? What that really tells you about it? Okay, this is a ribose. Okay, different places. CH2, OH, OH, H, H, OH, OH, H. Carbon number 1', 2 prime, 3 prime, 4 prime, and 5 right here, and 5 prime. Okay, what means 2 prime deoxynucleic acids? This guy is gone. Otherwise, it's ribose. So that's why deoxynucleic acid or deoxyribose. Okay? This is the first thing you need to know. Okay? Second thing we want to mention. What are the gene base of a DNA? Do you know the gene base of a DNA? What are those bases? What is A? Adenine. Okay. What is G? Guanine. What is C? Cytosine. What is T? Thymine. Okay, what are they looks like? I ask you to draw ATP structure. We're not going to draw all of them, okay? What are these two looks like? There is a 6 carbon with a 5 carbon. How about cytosine and the thymine? Just a very simple 6 carbon. Okay? So what's the first one called? Purine. What's the second one called? Pyrimidine. Is that right? So that's their gene base. Now you always remember what they combine together. What are the gene corresponding policy? A complemented with T, G complemented with C, and what's the name of this center bond? Hydrogen bonds. And the hydrogen bonds. But by the way, what are the 3 bonds of the chemical? What are 3 bonds? COAVELENT. Covalent bonds, ionic bonds, and the hydrogen bonds. Which one is most weak? Is hydrogen bonds. The weakest one. That's why these could be hydrolyzed. These could be hydrolyzed in a second. If these are covalent bonds, it's not going to be weak at all. Okay? And also, when you hydrolyze, the AT have 2 hydrogen bonds, GC have 3 hydrogen bonds. So more energy is needed to separate the GC. So therefore, when you design a primer of PCR, some of you do the research with a professor in some lab, you need to think about the GC components, the GC amount, because it's 3 bonds. Okay? So this is the second one is a base. Okay, third one, what is that? What are the second thing most important for a DNA? Think about that. Is that phosphates? Okay, so the second thing is triphosphates. Okay, triphosphates. So what it looks like. This is I'm going to draw a little bit here. So we have this guy here. Okay, this is H. This is going to be a base. Okay, then you go here, CH2. Then you're going to connect it with what? With oxygen, with a P, H, O, O, and then go here with a P, O, H, O, and here with a O, P, H, O, and OH. Okay. The triphosphates are very important. And this is the one we call it 5 prime, and this one we call it 3 prime. And how they connected with each other. Here you have OH, is that right? So what they do, they cut this line, so they're going to connect it, another one go here. is O goes P, then goes CH2, goes here, and this is the base, and then goes H. So what they do? Go this way, is that right? 5 prime to 3 prime. Now what are these bonds named? That's called the phosphate diester bonds. Okay? This is the 3 things important for the DNA. And you can read about these. You know genetics or no, A, G, C, T, A complement with T, G with C, and then blah, blah, blah. Then this is what I separate them to let you understand some important thing for the DNA. And this is already talked about. A and T, 2 hydrogen bonds, G and C, 3 hydrogen bonds. DNA running in opposite direction, 3 prime to 5 prime, and 5 prime to 3 prime. Okay, running opposite direction. Always remember that is 5 prime right there, connect to a phosphates, and the 3 prime goes here, which is a hydroxy base. So what this picture really tells you, what means they are running in opposite direction. Look at the sugar. The sugar is upside down. So the DNA strands running opposite direction. At the end of the day, is we are talking about is the sugar, the 5-carbon sugar, the 5-carbon deoxyribose is running in the opposite direction. Okay, so that is important. This is another one. If you think this is too complicated, let's look at this. This is the DNA polymer of nucleic acids, adenine, thymine, cytosine, guanine. Backbone is deoxyribose phosphates. A and T, G and C, they're running anti-parallel. Okay. DNA replication, we already talked about. It is semi-conservative. Melson's star confirmed it. It's semi-conservative. Okay. Then we're going to talk about the detail. And everything happens, which is a replication fork. And at the beginning, they are using E. coli as an example. Because the E. coli DNA is usually a circular. And then they started the replication. When they see the final terminology, which is TER, the replication will be in. So we're gonna spending a time, the rest of this class, about 20 minutes, we're gonna talk about the components of a replication fork and what really happens here. Okay. I'm going to left this one right here. This is important. We need it. I hope we have enough space. If not, we'll just move around a little bit. So again, you already learned, but I would just want to be, from my standpoint, so explain these to you in a different way to make sure you understand. That's my goal. Okay, first of all, there is 2 lines of the DNA running anti-parallel. Or just right here, okay, anti-parallel. Double helix. And A complementary with T, G complementary with C. Okay. At the beginning, one of them is 3 prime to 5 prime. Another one is 5 prime to 3 prime. Okay, when they started to have replication, everything that happened is right here, called replication fork. Okay, once this replication fork formed, they need to rely on a group of the chemicals, which is called DNA proteins. What are they? This is called the DNA A, DNA B, and DNA C. What is the DNA? Originate. DNA protein is originate the replication, which means give you a signal, it starts a replication. Okay, it's a double helix, highly twisted antiparallel. We need something separate. So what is DNA B protein? Helix. This is going to break down these double bonds. Okay? So you're going to break down these double bonds. Now what is DNA C? What does DNA C means? They need a holder or helper of helix. See, it's a double helix. When you see helix, of course it's going to separate. Okay, so once you separate, you need to make sure they are separated. Because they're always twisted together for a longer time, then from a physics standpoint, they're going to always keep it, come back. It's called tendencies, all right? So you're running, you suddenly stop. You're still going to have, kind of going forward, maybe fall. It's the same for the bacteria. So they're always twisted, they're going to keep twisted. So we need to make sure there is something there to stick to the two strands, separated, make sure they're separated. This is called SSB, single strand binding protein. So what's the function of these? They want to make sure this guy and this guy are separated. Okay, this is the initiation goes in. Then they start to do the replication. Okay, replication, how they start? Let's talk about 3' to 5', first strand. Okay, I'm going to give you something. Let's say the sequence is AP, TP, GP, CP, CP, GP, and TP, let's say. Okay, in this strand, we, based on the gene complementary policy, we're going to have to synthesize the complementary gene. Now, for everything started, initiate replication, they all need something. For DNA replication, they need